Thanks for joining us this evening for tonight's RTUK talks. I'm Lindsay and I'm the project manager for Radiotherapy UK. And our theme for this year's webinars is the Ambassador's Choice. We've got one of our ambassadors presenting tonight, this evening, and that's Dr. Sam Terry. She's a reader in radiobiology at King's College London, and she's also the founder of RadLab. Just a bit of housekeeping first. Please use the chat function to introduce yourself if you wish, and then for any comments and questions as Sam goes through her presentation, it's a good opportunity to engage with others and engage with the speaker, Sam, so we can have a question and answer session at the end if there are any questions to put to Sam. We'd also appreciate it if you have any comments to put in the chat for your main sort of takeaway from the talk. That'd be really good. And finally, we've got another RTUK ambassador with us, Katie Cooper. So big thank you to her. She's supporting us tonight and will be handling the chat. So thanks, Katie. So without further delay, I'm going to hand over to Dr. Samantha Terry, and she's going to tell you a bit about her work. So I'm going to stop sharing and hand over.
And I'm hopefully going to start the talk. There we go. Let me know if that's worked or not.
Yep.
Yep, super. So hi, I'm Sam. I am indeed a reader at King's College London, which is one of these weird academic positions where you're not a junior academic, as in like a lecturer status, but not professor either. Weirdly, my kids seem to think I just spend time at the library because I'm called a reader. Anyway, I'm here to, I guess, talk about the work that myself and my team have been doing over the last lots of years around radiation therapy. And yeah, as an academic, we've been doing research in cells and in animal models. I kind of wanted to describe a range of the kind of things that we do and have done in the past, and I guess what that might mean for the future in terms of clinical radiotherapy delivery. So I'm based at King's College London, which is this building. Can you see my cursor? No. Let me see if this will work. Laser pointer. There we go. It's this building just here. So very nicely situated. And yeah, I set up the team in 2015 when I got my academic position at King's, having done a PhD and several postdoc research positions, which are all like about 2 or 3 years in length before that. And yeah, we work with radiation, like I said, preclinically, and we work with all sorts of different types of radiation to really try and understand how our cells react to them, whether they're healthy, normal cells, or whether they are cells that have turned, I guess, cancerous. And we work with X-ray radiation. But we also do an awful lot of work with radioactivity, and as such, we get audited an awful lot simply because of the kind of work that we do, but also because of our location being in the tourist centre of London and also opposite the Houses of Parliament. Apparently, they don't like people wandering away with radioactive sources, so we do get checked quite heavily. I thought I'd start by giving you a bit of an inkling of who the team is. So these are some of the guys over the years. Many still are actually with us, and others have completed their PhDs and their postdocs and moved on to other research projects. And yeah, we're a mixed bunch. So I'm a biologist originally, but we have for example, Irene, who is a PET center, so radionuclide imaging technologist, who also is doing a PhD with me at the moment. She's halfway through at the moment. We have other biologists. This is Tian. She's a cardiovascular surgeon who's doing a PhD with myself and others. And then we have other people who are pharmacists, who are chemists, physicists, medical physicists, and yeah, all sorts of different backgrounds kind of come and join the team. And that's really because it is a very interdisciplinary team. And I know that that's an overused word, but it really is very true. We all come from very different angles. And the reason why I guess I've kept up, I guess, my enthusiasm around doing the research that I do is because of this number 1 and 2, which fortunately is still the number of people who are diagnosed with cancer at any one point in their life. But it's also the number of cancer patients who are being treated with radiotherapy. And of course, the majority of radiotherapy is delivered with X-rays. This is an extraordinarily simplified description of X-ray radiation on the left-hand side, defined here as external beam radiotherapy, and on the right-hand side, radionuclide therapy, which goes by all sorts of different names, such as molecular radionuclide therapy, targeted radionuclide therapy, radioligand therapy. It just continues. We have now settled as a community, I guess, on a name, which is good. And yeah, so the idea, I guess the big difference between X-ray radiation and radionuclide therapies is that X-ray radiation obviously gets delivered. It's still in a targeted fashion with multiple beams and conforming around the tumor area, but it's very different in the sense to radionuclide therapy, in the sense that you really can only target and treat what you see, whereas with radionuclide therapies, we're looking at injectable radioactive therapies that are made to target cancer cells that, when you inject them into the bloodstream, basically bind to your cancer cells wherever they may be, whether you see them on a scan or not, and irradiate your body from the inside out instead, also in a very targeted fashion, but slightly differently. And we've been doing some work very recently in head and neck cancer using X-ray radiation and combining X-ray radiotherapy with immune checkpoint inhibitors. And the idea here behind immune checkpoint inhibitors, this is all the rage at the moment when it comes to radiotherapy research, is how do the two mix, and for which conditions do they mix? And the idea here is that normally you have your cancer cell, as depicted here, that is in close proximity to, for example, immune cell, in this case, a T cell, and the two bind. The T cell has recognized that this is a tumor cell, normally an immune cell would get rid of anything in your body that isn't meant to be there. And that's what would normally happen here, except that the tumor cell has basically been able to overexpress this protein called PD-L1, which binds to this PD-1 protein on the T cell, and it stops it from working properly, and it stops the immune cell from getting rid of the cancer cell. So there's been a whole spate of different types of work and research into how do we stop this interaction, and there are loads of different inhibitors that stop that, which then enable the T cell to do its normal function, which is obviously get rid of a cancer cell. And what we, I guess, had a few years back is we had a clinician who works at, he used to be at Oxford, then came to King's, and now works at Guy's and St Thomas's Hospital as well. And he basically came to us and asked us, how can we possibly know and predict, I guess, which patients in the head and neck cancer area might benefit from combining X-ray radiotherapy with immune checkpoint inhibitors? Because what he was seeing is that, it would work for some patients and not for others. And he also noticed that an awful lot of people didn't have a strict regimen that they would stick to, and everybody just did things slightly differently. So we started working in this particular area and working preclinically. So we used mouse head and neck cancer cells. These had been created as naturally, I guess, as possible to mimic what would happen in the formation of a head and neck cancer in a human being. And we used some of these new cell lines, which are called very nicely NTCQ1, MOCL1, and MOCL2. They always roll off the tongue like this. And we irradiated them. And what we see here on the left-hand side is basically data from cell work showing that the head and neck cancer mouse cells respond to radiation. We can blast them with X-ray radiation, and we can see the formation of these green dots in this particular cell. The cell is blue because we've given it a dye to stain the nucleus of the cell. So here we're seeing a nucleus, and then inside that we see these green dots. And each one of these dots corresponds to a double strand break. So basically part of the DNA that has been damaged, and it's known that any unresolved double strand breaks will ultimately kill that cell. So the more green dots you have, the better chances you have of killing that cell. And we found that basically we can increase the number of green dots of double strand breaks when we irradiate our cells, and that even 24 hours after irradiation, they still have lots of unrepaired DNA damage, which is great. And this led to a decrease in the ability of these particular cells. So in blue, we have the MTCQ1s, and then we have the other two cell types in the other colors. But we have a decrease in the ability of these cells to survive post-radiotherapy. So this is super. We've got a model that is derived from mice, which means that when we put it into an animal model itself, that we can use an animal model that has an intact immune system, and we can start playing about and seeing what radiotherapy plus immune checkpoint inhibitors actually does. We could grow these cancer cells in the animals. So what we do in a preclinical study is we tend to inject cancer cells in a very small volume, usually on the flank of an animal, so that it doesn't interfere with any movement and it has as least of an issue with it as possible, so that it starts growing a little bit, I guess, like we might have a mole growing on our body. And we can monitor how much that tumor grows, and that's what we're depicting here. You can see that the blue line kind of shows a nicer increase in tumor size as time goes on, which is what you want in a preclinical cancer model. You want something that's reproducible and robust. And then what we did is we took those tumor tissues and we sliced them like you would slice a bit of ham, perhaps, or a bit of cheese, and we basically slice them and we stain them with antibodies for immune cells. And in this case, we stain them for, in red, we have dots that demarcate where macrophages, so a certain type of immune cell, are located in these tumors. And in green, we basically stained for T cells. And you can see that in this mouse model of head and neck cancer, you have the localization of both types of immune cells, T cells and macrophages, and that they have a very distinct. So what we found was that macrophages are basically on the outside of a tumor cell, and T cells were very much in the center. And then what we did after that is we irradiated these tumors to see what happened to that population of cells, and they started mixing, which was a really good sign for us, suggesting that, one, there was a response to radiotherapy in an animal model, but also that we weren't killing off all of our immune cells, so that they could have that positive anti-cancer effect. And then what we did, having decided that we were good to go with an animal model for basically mouse head and neck cancer, is we irradiated them, like I said that we had done. We basically have something called a preclinical precision X-ray irradiator, which is also image guided, so it's CT guided. In short, though, a much better term for it is an animal LINAC. And this is kind of what it looks like on the inside. So here's our X-ray tube, and then the animal would lie down here anesthetized. And we take CT images, and we would get, I guess, dose deposition maps. And we would work out our treatment plan, I guess, in slightly an old school way, but sometimes preclinically we're a bit behind with what's available in the clinic, and we set the treatment regimen to give us the maximum dose to the tumor and to the isocenter of the tumor, with minimal dose to the healthy tissues, such as here. And we tend to do it with two opposing beams at 180 degrees of each other to make sure we get the best coverage. And then we set up several different types of animal studies. I'm just going to show the data of one here, which is where we inoculated our cancer cells. So we injected them on the flank of a mouse. We waited till they were 100 millimeters cubed, so quite small. But then these are also animals, so we can't go too big. And then we had different groups of mice. So we had those mice that were left untreated and just had regular CTs. Then we had mice who were given 2 grays across 6 fractions, so a total of 12 grays. We had animals that were just treated with the anti-PD-L1 inhibitor. And then we had animals where we mixed radiotherapy with anti-PD-L1, but we either did it concurrently, so we started them sort of at the same time, or we did anti-PD-L1 treatment after radiotherapy was complete to see which would be best. And then we followed tumor growth, which is this graph on the left. And what we see is that control untreated tumors, they grow, which is not unexpected. When we treat with anti-PD-L1, we get some kind of delay in that tumor in terms of growing. And it's kind of the same with the X-ray radiation, which is kind of hidden under here. But basically the best inhibition, so the longest time that it took for tumors to grow was really in the group that received 2 gray of radiotherapy, then followed by anti-PD-L1, rather than giving the two at the same time. And this was also seen when it came to looking at how many mice survived. So in animal studies, we have to follow incredibly strict ethical rules and humane endpoints, and the size of a tumor or how a mouse might be behaving or looking very much determines if we have to stop the experiment or not. So that's where we're at with X-ray radiation. Like I said, we do an awful lot of work with radioactive therapies as well. And I just wanted to show this video. Apologies for the lift music. It's not my video, but I've never found a better version of this. So I'm going to play it now. So this is radionuclide therapy. It is a liquid. I'm going to remove the volume a little bit. So it's a liquid, and this is an example of a radioactive therapy that is called octreotate, which binds a receptor found on neuroendocrine tumors as well as neuroblastomas, and also a few other cancer types. And it has a chelator, like a cage, that binds lutetium in this case, which is a radioactive element. As a radioactive element, you get the emission of lots of energies, in this case a gamma ray and a beta particle, and it's the beta particle that's able to stop a cancer cell from growing. So this is the emissions, but of course, at the moment it's still in that liquid, or in this case a syringe, so it gets injected intravenously into a patient. As such, it goes through the bloodstream to wherever blood can reach. And of course, tumors are incredibly well vascularized, because that's how they get their nutrients and their oxygen to grow. And so our radioactive elements, which we have made to be cancer specific, get internalized. They go inside a cancer cell specifically, and obviously are still radioactive, so still releasing their energies and damage. And so a cell might die, and of course, when you look at a tissue level or a whole tumor level, that whole tumor will end up shrinking, as you can see here. And what's quite nice about these radioactive therapies, which is a little bit different to chemotherapies and other drugs, is that you can image where it goes. So you can get a really good idea of what your radiation dose is. And ideally, well, in most cases, we'll know that, and we'll be able to predict that beforehand, before we give the radioactive therapy. And yeah, there's an awful lot of mileage in radioactive therapies, and I could talk about that for hours, but I've been told I'm not allowed. So let's crack on with the next slide. There we go. So yeah, lutetium dotatate is the radiopharmaceutical that we just kind of looked at, and it's used in neuroendocrine tumors. And there was this trial called the NETA trial that showed that there was no increase in overall survival of patients that had been given this radioactive therapy versus the control group, but there was a very clear increase in progression-free survival in these patients, showing that it was worthwhile having given it in the first place. And what's happened now is we have a good understanding, I think, of what's happened here, and that led to, I guess, this lack of benefit when it came to overall survival. And this lutetium dotatate is now being moved earlier into the treatment pipeline, I guess, of patients, which is great, because up until this point, radioactive therapies were really only given when there was no other treatment available to a patient anymore. And it really is a booming market at the moment. So there are a lot of companies being created, spin-off companies being created, big companies like Novartis, AstraZeneca, GSK, who are dipping their toes into this market because they see a huge potential in terms of benefit in the clinic. And what we can do is we can, like I said, we can target radioactivity to cancer cells, and we can make it go to different locations, even within that cancer cell. So we can make it go on the cell membrane, we can make it go into the cytoplasm, we can make it go into the nucleus of a cell if we need to get it really close to the DNA. And it kind of depends on the type of radiation that's being emitted from this radioactive element as to where we might want to place it. Because some radioactive elements, like the beta particle, in the case of lutetium that we saw earlier, they might travel about 1,000 cells away from where the decay happens originally, so that you'll be irradiating at a very low level, similar almost to X-ray radiation, that distance of 1,000 cells in any direction. But then you have other radioactive elements, like those that emit alpha particles, such as radium-223, for example, or lead-212, that really create a more dense ionizing event in a smaller area, meaning that you might be irradiating about 5 cells rather than 1,000 cells away from where the decay happens. So it's much more localized, but also there's more energy being deposited, meaning that you end up having more complex and just more damage to your DNA in the first place. And then you have these other ones at the bottom, which are called Auger electron emitters. These aren't yet in the clinic, unlike the other two, and they're also not. There's one clinical trial just starting with Auger electron emitters, there's a terbium-161 that's starting at the moment, which we're all waiting with bated breath to see what the result is. And the Auger electron doesn't travel far at all. It really only travels nanometers, so the width of a DNA. But you end up getting not just one electron being emitted per decay, but potentially loads of them. So you still end up with a really high ionizing or ionized area. And you're really only irradiating the cell to which it's attached, which means it's incredibly exciting to look at Auger electrons in terms of maybe being able to target those metastases, so those small clusters of cancer cells. And that's because if you're using a beta or an alpha particle emitter, you might be missing. So you might have your radioactive element right in the middle of your metastasis, but most of the energy is being deposited outside of that. Whereas with Auger electrons, like I said, you're only irradiating the cell to which it's attached. So we might be in a good position to get rid of those and turn that progression-free survival increase that we saw for lutetium into actual overall survival benefits for people. And so we had a look to see, so to really make the most of radioactive therapies, we want to know what radiation dose we're giving and what's needed to give us 100% cell killing for the cancer cell. Obviously, dose limits exist both for X-ray radiation, but also for radiopharmaceuticals. But for radiopharmaceuticals, as I just described, they act very differently. They get distributed in the body differently, and their energy deposition is different to X-ray radiation. And so some of those dose limits are still based on what we know cells can handle, and kidneys can handle, bone marrow can handle when it comes to X-ray radiation, but it's being imposed on radiopharmaceutical therapies that are being administered at the moment. And that's a bit of an issue, because what we've seen, and there's evidence for, is that that's limiting us, and we're underdosing patients and not seeing that benefit as clearly as we otherwise would. So my team have taken a bit of a step back to really try and understand, what does the gray mean? So in terms of absorbed radiation dose, what biological effects happens? And so we use these breast cancer cells that we engineered to express this membrane transporter called the sodium iodide symporter. And that enabled us to add loads of different types of radionuclides into cells in a controllable way that we otherwise weren't able to. And we basically grew these breast cancer cells in animals and imaged where our radioactivity went. And we tested loads of different types of radioactivities, those that emit Auger electrons, those that emit beta particles. And we also looked at X-ray radiation. So we grew these tumors, injected the animals with radioactivity. The radioactivity went to the tumor. In this case, it's in the mammary fat pad. So basically just in the fatty area by the nipple of the animal, in where you would expect a breast tumor to be located. But you also see uptake in the stomach and also in the thyroid, and that's because they too express the sodium iodide symporter. So you would naturally expect it to go there. But we took loads of images over time of various different radionuclides to see how much was being deposited where over time. And then we worked out what radiation dose would be delivered to that tumor specifically. And then what we did is basically looking at tumor growth inhibition, where we delivered the exact same amount of gray of radiation to that tumor and looked to see how well it grew. And in untreated tumors, they grew quite rapidly, if not somewhat erratically. This is because, like I said, we sometimes need to take animals out of the experiment for other reasons. If we treated the tumors with X-ray radiation with our animal LINAC, we get very good control of that tumor. It's not growing at all. And if we inject either an Auger electron emitter, like in green, iodine-123, or in purple, dotty, rhenium-188, that's a beta emitter, we're able to still inhibit the growth, just not as well as for X-ray radiation. And this was also seen for our animal survival plots. But you might think X-ray radiation wins, and it does when it came to controlling the primary tumor. But in actuality, we saw an awful lot of metastases in the animals that were treated with X-ray radiation. And that's really because we only treated what we saw. We could only irradiate the isocenter of the tumor and nothing else. Whereas with our injectable radioactive elements, with rhenium-188, we saw far less metastases being formed, but still some. So in 40% of our animals, we still saw some in the liver. But when we irradiated with the Auger electron emitter, so the one that really only irradiates the cell to which it's attached, we saw no metastases whatsoever, which kind of shows that there is merit in still looking at these kind of radiotherapies, and that they can't replace X-ray radiation or any other treatment, but they are a very nice addition to that radiotherapy portfolio. So I wanted to describe that, and then two more small things. One, like I mentioned before, we have Irene, who's working with us, who works in the Nuclear Medicine and PET Imaging Center, working with radioactivity. And as a technologist, she is responsible for drawing up those syringes of radioactivity, but also injecting patients with them and following up the patients during their scan and afterwards. And as such, they receive she and her colleagues receive, a radiation dose of approximately 0.1 millisieverts as it stands at the moment. And she approached me a while back and said, look, we're concerned as a community that we're getting a radiation dose and we're not. And maybe that those dose limits aren't correct for us, whereas they might be correct for people being administering diagnostic X-rays or image-guided surgeries and things. What can we do about it? I've had a look in the literature. I can't find any data. And I said, well, why don't we just look into it? So we now have lots of blood samples of technologists who have been working in the field for varying numbers of years. And we also have blood samples before and after administering a high amount of radioactivity in the form of 18 FDG, which gives you the sort of images that you can see here on the right-hand side. We're just looking to see what kind of markers of damage there might be in the lymphocytes, the white blood cells, and those blood samples of people who've been working in the industry for quite some time. And I don't have the data, so I can't show you, but that's the kind of work that we can do, which is really quite nice. And what's most nice about this, I guess, is that it's come from the community And yeah, she ended up, she's still working as a technologist part-time, but doing a PhD part-time as a fellow. So she ended up getting a fellowship and is paid for that, which is quite magnificent. And then lastly, I just wanted to say, just like with Irene's work, it's very much driven by her. There's a large part of my work where about 10, 11 years ago when I started, it wasn't seen as part of my role. And now I've kind of made it part of my role, is this thing of public engagement and also patient involvement. And these are some of the guys I've worked with in the past and some of the activities I've worked around. But I think it's incredibly important to, when it comes to basic research, which is obviously the level that I'm at, and I don't interact with patients on a regular basis, I think it's incredibly important to know what's important to them, and also to know what the unknowns are for them when it comes to receiving radiotherapy. So we put a call out a fair few years ago now for young adults who had experience of radiotherapy as part of their treatment. And we ended up having patients who were treated with radioactive therapies, patients treated with X-rays, with protons, and yeah, everybody was so we've been working as, or collaborating together, I guess, for the last 4 or 5 years now, and it's been seen as quite worthwhile on all our parts. They've given myself and other researchers a lot of insight into what's important to them, which, you know, they came up with these top 10 tips for professionals delivering X-ray radiation, which has been published in the British Journal of Cancer and has been highlighted at many different conferences like ESTRO and places. Yeah, the key thing, I guess, especially seeing things I have you guys here, especially if you are providing radiotherapy treatment for patients who are slightly younger and are considered young adults or teenagers. The key thing that came out of that was just don't overlook us. Treat us as people first, not patients. And yeah, there was an awful lot around in our discussions around side effects and long-term side effects, and also some of the negatives around receiving radioactive iodine for thyroid treatments. And yeah, there's a lot of learnings. And like I said, I could talk for hours on this, but regardless of where you are at as a researcher or as a professional in this area, there is always a good reason to talk to people, even if it's just because it's the right thing to do. And sometimes people need listening to, or need to unload, or need to understand what happened to them, or why they're having X, Y, Z, and what, you know, we were able to explain a lot about certain symptoms that happened, or what was the behind the scenes, or how did people feel delivering X-ray radiation, rather than how it was perceived for people who received X-ray radiation therapy. So yes, a lot to be said on that one. Right, so key thing is, as a radiation researcher, me and my team, we cover a vast range of work. This is just a snippet of the kinds of things that we do. We've been blessed with a lot of research funding over the years from various different locations. And yeah, just kind of wanted to end on this slide, which just described, I guess, the biggest thing that we did as a team of researchers and patients, where we discussed, I guess, what their radiotherapy experience over the series of 3 workshops. Also on they were Wednesday nights, but also around this time of the evening. And yeah, these were the outputs. So thank you very much. That's me.
Amazing. Thank you so much, Sam. That was quite fascinating. Blew my mind, actually, some of it. And I didn't realize quite how effective, radionuclide therapy can actually be. So
Yeah.
It's quite an eye-opener, actually. We do have a couple of questions. One of them is, do you know what diagnosis King's College are treating with radionuclide treatment at the moment, and what volumes?
I don't know the volumes. In terms of treatments, we're looking at thyroid cancer. So with iodine 131, we're trying to get prostate cancer patients. That's a level above our trust. That's to do with, I guess, the ability to get reimbursed for that. Patients with neuroendocrine tumors are being treated with lutetium dotatate. And then across London, you've also got, so at UCLH, I guess you've got neuroblastoma patients being treated with iodine 131, MIBG, and also sometimes lutetium dotatate. Yeah.
Thanks,
But
Sam. This was a great talk. I'm joining you from Canada.
Hi.
And
It's a lot earlier.
yeah, it's much earlier. You've probably all had your tea.
Yes.
Yeah, in Ontario, where I am, they've just loosened the criteria for prostate. It had to be 2 failed treatments before, and now they've moved it up. And similarly, at Princess Margaret, London, Ontario, a couple of centers treating the same diagnosis that you just named, but we're finding across the North America, there's a lot of work being done on prostate, and Novartis is a big player. But
Yeah.
thank you. I love that slide where you have the picture of the difference between radiation and nuclear radiation. That's, for those who may not understand, that was a great slide. But thank you.
I'm glad. I realize it's an oversimplification on both sides, but yeah.
Perfect.
There are some really exciting clinical trials ongoing at the moment, in particular for glioblastoma, so brain conditions, looking at Auger electrons, and then being able to treat there. So I guess in a similar fashion as you know, proton beam radiation is very useful for that kind of condition because of the limiting damage to healthy tissues. The same is thought that could be possible with Auger electron emitters, and that might be a bit more. We might be able to treat more patients in that way rather than, because obviously there aren't many proton beam centers. So
yeah, no, that you've only got 2.
yeah. I mean, we got 2 though, so this is a good start.
Better than none. No, it's fascinating what's evolving. I mean, I know radioactives have been around a lot. I'm a nurse, by the way. But what's evolving, it seems to be very, very exciting. And in terms of your program, sorry, Lindsay, if I'm asking too many of the questions.
We have time.
In terms of your program, obviously nuke medicine plays a role, radiation therapists. Who are actually giving your infusions? Because that's where we find the workflow is complex, and I call it's often built on the yellow sticky note program.
So who delivers it, as in like physically delivers?
Mm-hmm.
So it'd be people like Irene, but of course it's backed up by a massive team, just like in X-ray radiation, right? In terms of physicists, etc. Well, radiopharmacists and people too. Yeah.
In the North American market, it'll often be the chemotherapy nurses who are.
I see.
Yeah.
Wow.
Yeah. So that means you have another stakeholder in the whole group, which means more coordination in terms of getting it all done. Yeah. Thank you.
Really.
No, thank you. I have one more question. How long does it take to get from the labs, from these preclinical sort of trials into the clinic? Like, what sort of time scale? Or is that, how long is a piece of string?
It really depends on how new the radiopharmaceutical is, and also where you're trying to do your clinical trials. So in the UK, it's very difficult to get clinical trials on the go for this kind of thing. So a lot of companies, even if they're UK based, end up going to Australia or Germany.
Yeah.
Yeah.
Okay.
So which is good in the sense that it expedites
Gets things done quicker.
Exactly, it gets things done, which is in the long term better for us, even in the UK or other countries.
Yeah.
Absolutely, but yeah, it depends. So you know,
It's quite
if you wanted to,
frustrating, isn't it, really?
it very much is. We're working on it as a community, but there are lots of hoops. But if you've got a targeting vector like dotatate or PSMA, you're simply attaching another radionuclide onto it, then the barrier is much lower and therefore the translation to the clinic will be much faster.
Right.
Yeah. So what my team tries to do is to make sure that we have that biological understanding of what will happen and what kind of activities you need to get where to get that efficacy, which ultimately will help the speed at which other people make new compounds available. And also, if we know what, this is the other thing, if we know what level our kidneys can tolerate for this radionuclide or that radionuclide, that will hopefully allow us to administer the correct amount of radioactivity as well.
Yeah, it's quite interesting. I just have what I found quite fascinating when you were talking about what dose to give, and you're very much governed by kind of external beam radiotherapy and sort of gray and what we consider to be, you know, high dose for whatever area. Because I mean, at the end of the day, we're governed by actually the normal cells, aren't we? And what damage we're causing to the normal cells. We're not kind of governed by the tumor cells and how much damage we can cause them, in a sense. So it's completely the opposite, in a sense, to what you're looking at and what you're wanting to achieve with radionuclides, I think.
Well, so we're also governed by healthy tissue toxicity, but what's different is if you irradiate a kidney, for example, with X-ray radiation, or the heart, let's take the heart, because that, I guess, happens more often than a kidney. But if you irradiate a heart with X-ray radiation, you're not you're getting not a uniform dose across, but it's more uniform than if you were to use a radioactive source. If you know, in a tissue, you would, if you had a radioactive source and it doesn't really localize in the heart, so it's quite a bad example. But it will go through the urinary tract and get excreted via the kidneys. So it would localize in the kidneys in most cases. And then, you know, if it just goes through the kidneys and out, not a problem. But if it stays there, then potentially it's a problem. But it might only be a problem if you use a certain radioactive element versus another. Because if you're using like an Auger electron emitter and it doesn't travel very far, then you might kill one cell, but it won't change the organ as a whole and its function as a whole. Yeah, so there's other things to think about, but the fact that that dose limit is so set on X-ray radiation and some iodine-131 data, it makes it difficult for new things to come in and be used at the right level as it stands at the moment.
Yeah. Wow, it's a big subject. Amazing. Thanks so much, Sam. That's just yeah, amazing stuff. I'm gonna move on now because that's all of our questions. And if you're able to stay with us, then I'm just going to give you one update from the charity Radiotherapy UK before we say goodnight. We've got our Mars Radiotherapy 2025 challenge that's open now, ready for sign-ups. This year we've gone all techie and we've joined forces with Race at Your Pace app. And it's a really funky little app that means you can compete equally. So even if you're part of a small team, you can compete and you can still be measured on the leaderboard because it kind of breaks it down into the number of people in your team. There's also a leaderboard for those that are competitive. And my favourite part of it is the multi-activity button, because I'm not a runner or a walker or whatever, but I do gym classes. And you can convert your housework or your gym class or your boxing into miles, and it does it all for you. And you can log those, which I think is great. So it's £20 to join in, and we're trying to raise money in the charity to make more videos and resources for our patient and families hub. And these are the things that basically patients and professionals have been asking us for. So yeah, just for me to say now, thanks for coming along. Really appreciate you being here tonight, and really hope you enjoy the rest of your evening.